Hello Year 10 Triple Science Biology class. This lesson is a recap on the nitrogen cycle. Your very last lesson that you had before Christmas holidays, we completed the work on the carbon cycle, the QWC response, and went on to start looking at the nitrogen cycle. So a little starter activity, it's only supposed to take a couple of minutes. So pause the video at this point and I want you to answer the two questions that are on the screen. Why is nitrogen so important? And even though 78% of the air we breathe is nitrogen, why can't we extract our nitrogen from the air? Pause the video and see if you can answer that question. So nitrogen is needed to help us make proteins in our body. We need nitrogen to help us grow and help us make new cells. So sources of protein for us in our diet are normally things like meat, egg, fish, chicken, nuts and pulses. We eat these and they get broken down into amino acids in our body. Our body then uses the amino acids to help us make the proteins that make up our skin, our muscle, our bones and other parts of our body. So any excess amino acids then get taken to our liver, they get turned into urea, and our body gets rid of that waste urea when we go to the toilet in the form of our urine. When our bodies die, decomposers break down the dead body and start to release some of the nitrogen that was formed in these protein structures in animals and in plants. Even though there's 78% of the air that we breathe, excuse me, we can't get nitrogen straight from the air because it's very unreactive. So in your booklets, excuse me, I want you to turn to page 19 for me, please, where you have this diagram. Now I'm hoping that most of it was filled in from our previous lesson, but I think we got right to the very end of the lesson and we need to finish this off. So I wanted to make sure that you understood it completely. So in our diagram here, we have got some cattle at the top of the page, we've got some plants on the left-hand side, and then it tells us the various forms that we find nitrogen compounds in, as well as nitrogen, the element. So if we start with the plants on the left, plants grow up, they need nitrates to help them to grow, and they get these nitrates normally from the soil. So if we start at this point just here, the nitrates that are in the soil are being absorbed by the roots of the plants. These plants then get eaten by animals, and the animals take the nitrates that were in the soil that passed to the plants and were made into proteins, and part of those nitrates then become part of the animal's body as in the form of proteins. When these animals excrete or when they die, they start to get decomposed and broken down, and so the proteins from their body, from the muscle, from the meat, okay, and also the urea from their waste, gets broken down by these types of bacteria here. They're called decomposers, and includes bacteria that we find in the soil. So decomposers are bacteria and fungi, and they break down the proteins and the urea from the waste and from the dead animal's body into ammonia and ammonium compounds. The ammonia then gets broken down by another type of bacteria, second type of bacteria. This reaction is an oxidation reaction. So in chemistry with Mrs. Lees, you will have learned about oxidation reactions. They are just reactions where oxygen is added. So one of the very first oxidation reactions you probably observed in school was when you burnt magnesium in a Bunsen burner flame. By burning it in the flame, the oxygen combined with magnesium to make magnesium oxide, and you got a bright white flash and some white powder left on the end of the tongs. So all oxidation is the addition of oxygen to another compound. So in this case, the oxygen gets added to the ammonia, and the ammonia becomes the nitrates in the soil. They often dissolve in the water so that they're present in the soil. Now this nitrate in the soil can turn straight back into nitrogen gas to go into the air by a third type of bacteria called our denitrifying bacteria. So they do the opposite of the nitrifying bacteria. They take the nitrates, remove any oxygen, and they turn them into nitrogen in the air. There's one final type of bacteria that you need to know about, and that's the nitrogen fixing bacteria. Now these are only found on plants that we call legumes or leguminous plants, the beans and the peas. And they are the only plants that can take nitrogen directly from the air and turn it into a form, where it can be absorbed by the roots. In other words, they turn it into nitrates. The same thing can happen actually by lightning as well. There's enough energy from a lightning storm to take nitrogen straight from the air. Remember, there's 78% of nitrogen in the air. And to turn it into nitrates, and then that dissolves in rainwater, in the clouds, in the rainwater during the storm, and then when it rains, that nitrate-soaked water goes into the soil. So what I would like you to do is see if you can fill in the table that's on page 19 to describe exactly what's going on at each part of this cycle. At the feeding part, so that's up here. At the death and excretion part, over here. Decomposition, here. Oxidation, here. The breakdown by denitrifying bacteria, here. The nitrogen fixing bacteria that are found both in the plants and legumes, but also in the soil and lightning. And finally, absorption by the roots. This way, if you learn it in this particular sequence, it helps you to remember things a lot easier. So, a couple of tasks now that are not in your book, that I would like you to have a go at. You can write these on the back page of your booklet. This is only a 5-minute task just to break up that quite heavy part of the lesson that you've just completed. I want to know what the 4 types of bacteria are that we've just covered, that we've just talked about in the nitrogen cycle, and I want to know exactly what each do. So this is a literacy task. So turn to the very back cover of your book and you can bullet point them. I want to know the names of the 4 bacteria and I want to know exactly what they do. When we're explaining this for a B grade, it's very important that we say what they turn the chemical that they're acting on into. So there's 2 chemicals, the chemical that the bacteria act on and the chemical that they turn it into. Pause the video and have a go at doing that task now. So hopefully you should have come up with, from the previous cycle diagram that we've talked about, if we're doing it in the same kind of order, we would start off with the decomposers. So the decomposing bacteria and fungi, they take the urea or waste compounds or the protein and they turn them into ammonium compounds. The second type of bacteria in our description are the nitrifying bacteria. And these are the oxidizers. They take the ammonia or the ammonium compounds and they turn them into nitrates. These are particularly important, these nitrates, because they can be absorbed by the plant to help them with growth. The third type of bacteria are the denitrifying bacteria. They take the nitrates and turn them straight back into nitrogen gas in the air. And the fourth type of bacteria are our nitrogen-fixing bacteria. What we mean by fixing nitrogen is they take nitrogen straight from the air and are able to convert it into nitrates that plants can take up or get absorbed into the soil. And as we said before, lightning also does a very similar role. Check your answers and make sure you understand the names of the 4 different types of bacteria involved in the nitrogen cycle and exactly what they do. Right, a little task here. This is a numeracy task, and again, this task is not in your booklet, okay? So, actually, I think it might be in your booklet. 1 second. Yes, it is. It's on page 24. Apologies, okay? The only thing I haven't included in your booklet is graph paper, so I do apologise. So you'll have to attempt a graph the best you can, either using graph paper if you've got some at home, or if you can, print some out, or just do it on some plain paper to judge your scale accurately. So this is about application of data. We've got 2 areas, area A and area B. In each area, there might be different types of soil. In other words, soil that might be nitrate rich or nitrate poor. We have a control area in each area, and we have an area where they've been given fertilizer and an area where there are known root nodule bacteria. So they might have grown, for example, some leguminous plants there, things like peas and beans and broad beans. I want you to look at the amount of seed produced. The kg/HA is kilograms per hectare. Okay, so hectare is an area, a unit of area, and kilograms is a unit of mass. So I want you to draw a graph to show the control, the fertilizer, and the root nodule bacteria, and the amount of seed produced in areas A and B. So pause the video to draw your graph. For the second part of the task, you can do this underneath on the lined paper that's provided on page 24. I want you to answer this question. Explain which area, A or B, and also which conditions, control, fertilizer or root nodule bacteria, provide the best conditions for seed production, to maximise seed production. Use the data to provide evidence for your choice, and this is not just a simple 1 marker. There are 5 marks to be gained here, okay? It's very important that you compare the data, okay? Not just for the 2 different areas, but also for the different conditions, the control, the fertilizer, and the root nodule bacteria. So have a go at writing your conclusion for that task. So this is what your graph could potentially look like. So the way it's been set out here is we've got the control for area A is on the left and B is on the right. The conditions with fertilizer, A on the left and B on the right, and root nodule bacteria, A on the left and B on the right. With each of these graphs, we can see that area B clearly gives us the most amount of seed. So there's not an awful lot of difference here in the control experiment. So the control experiment wouldn't have any additional nitrogen fertilizer or root nodule bacteria added. But when it comes to the fertilizer, but more importantly, okay, the root nodule bacteria, this increases it much more than any other. So remember what we said these root nodule bacteria do? They are nitrogen fixers. They can take nitrogen directly from the air and turn it into nitrates. Fertilizers also contain nitrates. Fertilizers can either be artificial or they can be organic fertilizers such as manure or chicken poo, okay, is often used, okay, to scatter on fields. So describe, remember you've got 5 marks, describe the difference between the control, and if you can use numbers and figures that will help. Make sure that you're using the correct descriptions. Make sure you describe the difference between the fertilizer, okay, and the root nodule bacteria. So your graph should have label on the y-axis, x-axis, apologies, sorry, this is the x-axis, this one. Label on the y-axis with the units, don't forget those. A title, accurately plotted bars, okay, precisely plotted to get full marks for your graph. So in terms of the evaluation then, area B is better than A. That would get you 1 mark. Why? Because it always produces more seeds than A. I should say that. Root nodule bacteria provide the best conditions for 1 mark. Why? Because they produce more seeds than either the control or the fertilizer. And look, this last mark is for you including correct use of data. So actually stating the difference between the amount of seed produced. So how many marks did you get? Write your marks out of 5. So here's our summary diagram to show the nitrogen cycle. Okay, make sure that you know the names of the different bacteria: decomposers, nitrifying, denitrifying, and our nitrogen fixing. There are 4 bacteria. Make sure you know what they act on: proteins and urea for decomposers, breaking them down into ammonia. Ammonia broken down into nitrates for nitrifying bacteria. Nitrates into nitrogen for denitrifying bacteria, and nitrogen back into nitrates for nitrogen fixing, okay? The important thing to remember is that there's protein in our plants when they take up the nitrates, and that the animals feed on this, either plant protein or other animals. And it's the protein in their body that when they die gets decomposed. Okay. The next task that I'm going to set you is for the second hour of this lesson. These are some practice GCSE questions. Now, there are a number of practice GCSE questions also in your booklet. Those you should make a start on. You should be able to attempt most of those. There are some that are carbon cycle, some that are nitrogen cycle, okay? And the one on page 21 is a QWC, which I think is where we finished just before Christmas, okay? So make sure that you could describe how nitrogen is recycled through the action of just soil, bacteria and fungi for that one. And also refer to the factors that affect bacteria and fungi during recycling. So think of our lesson on decay, that warmth, moisture, okay, and oxygen, the presence of oxygen and a source of food is going to help with decay. And then just a different style of nitrogen cycle question is on page 22. This one starts with urea in animal waste, and it wants you to explain how the nitrogen in the urea is recycled to become protein in beef. So we're starting literally from here, and it wants you to describe this section of the cycle. Back to our cattle again, okay? To help you, when you've marked that particular question, on the opposite page, I've put some success criteria. These are general ones for nitrogen cycle tasks. So not all of them may necessarily fit, okay, for your particular question. But it puts them into a sequence and logical order so that you can see. So you can use the peer assessment column to tick off what you've included in your questions for both the one on page 21 and the one on page 22. So the questions that I've got here on the board, these are actually AQA exam board as opposed to WJC, but they will be very, very similar. This one is about an experiment students did about decay of grass. So they took 200 grams of grass and they put them into 3 thermos flasks. All flasks had air holes in the lid. That tells us instantly that there's going to be oxygen able to get into the flask for aerobic respiration. We know that thermos flasks are good insulators of heat. They keep the heat in. And we've also seen experiments where we've had peas in thermos flasks back in our B1.2 unit in the double award lessons, okay? And looking at as they respire, they release heat energy. The diagram shows the conditions in each flask. So in flask 1, we have grass and disinfectant. So this should be familiar. We know what disinfectant does. It kills microbes. We have dry grass, no moisture in there, and we have wet grass. So think about the conditions we've just talked about that are required for decay. Each flask has a temperature sensor attached in the top of it, connected to a display, normally with a data logger to collect the data over a long period of time. So this was the data that they collected. Flask 1, if you remember correctly, that was grass and disinfectant. So this is with disinfectant. Flask 2 was dry grass, and flask 3 was our wet grass. Graph 1 shows the results given by the processor and display after monitoring the temperature through the temperature sensors over 5 days. Give one advantage and one disadvantage of using these temperature sensors connected to a processor to display and record the results. So have a go at answering these questions. I'm afraid they're not in your booklet, so if you take a piece of paper and write them down, or you can write them on the cover of your booklet, somewhere where they're going to stay safe. Second part of the question, compare the changes in temperature between flasks 2 and 3 over the 5 days. Now there are 4 marks for this question. So if you go back between flask 2 and 3, that's this one and this one over the 5 days. And you've got to get 4 marks for this. So think about what you're going to include. And finally, 1A part 3, explain the changes for flask 3 from day 1 to 5. So why do we get these results in flask 3? So have a go, pause the video, rewind the video if you need to have a look at the data, and see if you can attempt to answer them. Final part of this question. The temperature for flask 1 changed very little. Suggest why. So think about what we'd said about the disinfectant. And the following advice was given on a garden compost bin. Fill with organic waste such as waste food, peelings and plant material. Keep air holes clear and turn the material regularly. What is the reason for carrying out instruction number 3? And there are 2 marks here. Pause the video, answer these last 2 parts, and we'll go over the answers together. So, for question 1A, we've got 2 marks that it wants. An advantage and a disadvantage of basically using a data logger, a temperature sensor and a data logger. The advantage is that you get accurate readings, or that a person is not needed to measure the temperature, or the graph is drawn automatically. It's easier to read the information, it's quicker to process. You could even have that it can be left and set up overnight, okay? And you don't need to have a scientist there taking regular readings. Disadvantages though, it's expensive. It needs to be set up, it needs some technical knowledge to leave it running for a few days to collect that data. So it's good to be aware of this experiment, as it's recommended not just in the AQA spec but also in the WJC spec that you understand the advantage of using things like sensors and data loggers over regular things like thermometers and digital thermometers. So for 1A part 2, compare the changes. Now there are 4 marks here, but you can see in the mark scheme they give us a total of 6. So you only need to pick up 4 out of these 6 to score full marks. So you could say that the flask 2 temperature rises very slowly or only a little, and we can see that because it's quite a shallow line on the graph. Whereas flask 3 temperature rises by more or by 7 degrees Celsius. So if you can use figures from the data, that always helps. Flask 3 has a steeper increase. So it's looking at the shape of that graph, the angle of that graph. Whereas flask 2 has no steep increase. Both start at the same temperature or at 21 degrees Celsius. But flask 2 stays the same, almost the same for the last 3 days or from day 3 to 5. So it's really analysing that data in detail. Remember, these type of compare questions should have words like whereas in the answer to highlight that you are looking at 2 things, not just describing one then the other. You'll notice that there is a lot more to talk about than you might first think. Explain the changes in temperature for flask 3. So this part of the question, explain, is very important. First of all, you've got to get the idea that there are microbes or microorganisms that cause decay or rot the grass. That these microorganisms respire. That this releases heat and that moisture is needed to increase the growth of microbes. And more heat is produced over time because the microbes reproduce and divide and grow. Explain the changes in the temperature for flask 3 from day 1 to day 5. So again, 3 marks, and there's a total of 5 that you could pick up here. That the microbes decay or rot the grass, they carry out respiration, releases heat, increases growth of microbes, and they reproduce or divide. I've just realised that we've just said that a second time. Temperature for flask 1 changed very little. Suggest why? That the microbes were killed by the disinfectant. It's as simple as that. And why should we turn the waste material regularly? It mixes the microbes with the waste. It helps to release heat so it doesn't get too hot. And it also allows air to get in. You only need 2 of these for this particular question. So pause the video, you can rewind it, check your answers, see where you're missing marks, okay? What the little steps are that you're missing out that will enable you to get maximum marks. Last question, I want you to have a go at seeing if you can remember the carbon cycle. So this is from over a month ago that we did the carbon cycle. So it's right at the very beginning of your booklet. This is a QER question. So in this question, you'll be assessed on using good English. Excuse me. The diagram shows a very small part of the carbon cycle. So we've got carbon dioxide is given off by fossil fuels. The plants take up the carbon dioxide. The animals eat the plants and the animals give off carbon dioxide. So it's not as simple as just describing that flowchart as I've just done. That's not going to pick you up marks. I want a description of how carbon is cycled in the environment. You must include the key processes and how carbon is used in living things. Now, if you remember in your booklet, after we did the lessons on waste, on decay, there was a piece of text that you had to extract information from, and you highlighted things like all the places that the carbon is found in one colour, all the processes that take carbon from the atmosphere in another colour, and all the processes that return carbon to the atmosphere in a third colour. So that will help you, okay, to be able to answer this question. Take your time. I want you to spend some time planning it out. So on a piece of paper, you can do yourself a rough plan, just as you would in an English essay, okay, making sure that you've got all the key words. And then from your draft, okay, from your key points that you've made that you want to include, that you think will help you pick up maximum marks, I want you to have a go at writing your answer. If you can do this without using the notes in your book, then that's absolutely fantastic. It shows that you've understood it and you can explain it clearly. If you need to pop into your book and have a quick look at a carbon cycle diagram just to prompt you, then please, by all means, do. I don't want anybody sitting there struggling, thinking, I don't know how to start this, I don't know what the processes are. So it's been a month since you've done this, so this is really good retrieval practice. And I will go over the answers that the exam board would be looking for in a minute. Pause the video. So, with all exam boards with a QWC question, the first thing they would do when they read a student's answer is they would decide whether or not they're going to put it into a level 3, level 2, level 1, or no marks at all. The way we establish this, if nobody's written anything, it's generally 0 marks. If there's a simple description, but there's not very many key words, it would be in the 1 to 2 mark section. If there's a clear description of the carbon cycle and 1 or 2 uses of the carbon are mentioned, it would be in the 3 to 4. And if there are more points than just the parts shown. Level 3, to get the maximum marks, it's a detailed description with all the key processes mentioned and most of the uses of the carbon in the body of plants and animals mentioned. We'd also be expecting at this high level to see almost virtually no mistakes, spelling, punctuation, grammar to be perfect. Level 2, you might have a couple of spelling mistakes, you might have forgotten to put some capital letters, but you're starting to get the idea of a clear description. So what about the actual content? So most examiners will read through your answer twice. They'll be looking for whereabouts they think you fit in terms of the level, and then they'll be looking for the content. So, points that you could make is that carbon is used, normally most people get to make carbohydrate, okay? Some people will mention starch for energy, carbohydrate for energy, or sugars. Most people generally don't mention the cellulose, so if you can start to include that, that would be brilliant. Cellulose is required to help make the cell walls of plants. Carbon's also used, okay, to make protein for growth, and also fat for stored energy. So don't forget to mention those. So we're just going on to the unit on digestion. The booklets will be coming out to you the end of this week. So we'll be talking about the role of all these different food groups, carbohydrates, proteins, fats, and vitamins and minerals later. Carbon is found in the air in the form of carbon dioxide. Please do not be tempted to write formula in an extended prose answer. And if you are going to write formula in any answer, make sure it's correct. CO2 is written like that with the 2 as what we call a subscript. If you wrote CO2 like that, that is wrong. With the 2 superscript, that is the wrong way to write the formula for carbon dioxide. But please just avoid it. Write it out in full, okay? This is assessing your quality of written communication, not your knowledge of formulae. The carbon dioxide is taken in by plants, or you could say algae. I haven't seen that very often, but some people do. And the key process is for photosynthesis. The carbon dioxide is also then taken in by plants or animals that have eaten the plants, okay? You need to mention decay. Decay of dead plants or animals or waste. That could be the fecal waste, okay, or urea, as we've talked about in the nitrogen cycle. And you should be able to tell me that decay is caused by microorganisms or microbes or detritus feeders, so that's things like our fungi and our bacteria, okay? And that these things, these microbes, carry out respiration. Respiration is also by the plants and the animals. So plants carry out respiration during the night, animals carry it out all the time. And the final process is that fossil fuels, okay, from dead animals and plants that have been fossilized and turned into things like coal, oil and gas, when they are burnt, try to use the key term combustion, that this releases carbon dioxide. So it only requires a simple understanding of the carbon cycle, but you will get marks for knowing a little more. For example, use of carbon to make cellulose or carbon being locked away in limestone. You can get full marks for just the material in the specification, okay? Right, so make sure that you try to put as much information as you possibly can. So proofread your answer and you tell me how many marks you would give yourself out of 6. Thank you, Year 10.